This chapter is going to look at glycolysis, which is chapter 18 in your text. Okay, this is going to be the first metabolic pathway that we look at. So glycolysis is a sequence of reactions that metabolizes one molecule of glucose into 2 molecules of pyruvate, and we produce a net of 2 ATP. Okay, so let's draw the structure of glucose here. Okay, so here we have glucose. It's a 6-carbon sugar. Let's draw the structure of pyruvate. Now if you look at this, here we have 3 carbons. So we're going to take something that's 6 carbons and make 2 molecules of 3 carbons. And the net reaction for glycolysis is the following: glucose plus 2 NAD plus 2 ADPs plus 2 molecules of inorganic phosphate is going to give us 2 molecules of pyruvate plus 2 molecules of NADH plus 2 protons plus 2 ATPs and 2 water molecules. Okay. This, for glycolysis, you can see there's no oxygen. This process is anaerobic, and it's thought that it evolved when there was little to no oxygen in the atmosphere. Okay. We can take the pyruvate and it can be processed aerobically or anaerobically. Okay? It depends upon the reaction conditions and what state our bodies are in. When we process it aerobically, pyruvate will get completely oxidized to carbon dioxide, and you generate more ATP. When it's processed anaerobically, we make lactate through fermentation. And we get less ATP. Glucose is a common and an important fuel. In mammals, glucose is the only fuel that the brain uses under non-starvation conditions and the only fuel that red blood cells can use at all. Almost all organisms use glucose, and most that do process it in a similar fashion to the glycolysis pathway that I'll show you. Glucose is both an excellent fuel and a versatile precursor, as you can see from the figure here on the slide. It supplies a huge array of metabolic intermediates for biosynthetic pathways. In animals and vascular plants, glucose has 4 major fates. It may be used in the synthesis of complex polysaccharides destined for the extracellular space, okay, and that's shown here, where you're making the extracellular matrix and cell wall polysaccharides. It can be stored in cells, okay, and for storage, we store glucose as glycogen. It can be oxidized to pyruvate, okay, and that's what we're going to look at today. And so then this is where we can get ATP. And it could also be used to make other metabolic intermediates in the pentose phosphate pathway, and it's a precursor for ribose 5-phosphate. So why glucose? Now glucose is used as a prominent fuel instead of other monosaccharides for 3 main reasons. First, glucose is one of several monosaccharides formed from formaldehyde. Here's the structure of formaldehyde. And so, under prebiotic conditions, this glucose may have been available as a fuel source in primitive biochemical systems. Second, glucose has a low tendency relative to other monosaccharides to non-enzymatically glycosylate proteins. The proteins can be modified with sugars. And glucose has a low tendency to do this compared to other sugars. Finally, glucose remains in its ring conformation. Now, monosaccharides can either be open chain or in rings. When they are open chain, they often have either an aldehyde or a ketone. The aldehyde and the ketone can react with nitrogen, and you form a Schiff base, or if you remember back to organic, and we'll see this later, an imine. Okay? And so we don't want this reaction to occur. We would like glucose to remain stable in its ring conformation and only be in the open chain when we need it to react accordingly. So by the end of this lecture, we're going to look at the chemical basis and logic for glycolysis, and which is the central pathway of metabolism, and we're going to see how glycolysis works. So if you remember back to that metabolic map, okay, we are looking at this part here, which is glycolysis. Alright, and if we're thinking in terms of biochemistry land, we are in carbohydrate land. Alright, so this lecture should address the essential features of glycolysis. We're going to see why coupled reactions are important. We're going to look at the chemical principles and the features of the first phase of glycolysis and the second phase of glycolysis. We are not going to do the metabolic fates. Actually, I'll just change colors so it's clearer. We're not going to do the metabolic fates of NADH and pyruvate in this lecture, or the regulation of glycolysis. That will be for another lecture, which will be lecture 3. And again, we're going to skip other substrates and hypoxic stress for now. Alright, so we're going to focus on these top points here in this lecture. Alright, so what are the essential features of glycolysis? The glycolytic pathway is common to virtually all cells, both prokaryotic and eukaryotic. In eukaryotic cells, glycolysis takes place in the cytoplasm. Okay? There are 10 enzymes involved and 10 phosphorylated intermediates comprised of 3 or 6 carbons. And this pathway can be thought of as can be broken down into 2 stages or 2 phases. The first phase is going to result in the conversion of glucose to 2 molecules of glyceraldehyde 3-phosphate. The second phase is going to produce 2 molecules of pyruvate. We're going to net 2 ATPs and also make 2 NADHs. Right. There are 3 possible fates for pyruvate, and I'll go through them pretty quickly, and we'll explore them in more detail in another lecture. Okay, so first let's look at the first phase of glycolysis. Okay, no ATP is generated in this stage. Okay, stage 1 is the trapping and preparation phase. Alright, so again, no ATP is generated. We're going to begin with the conversion of glucose into glucose 6-phosphate. Okay? Then we're going to take the glucose 6-phosphate, make fructose 6-phosphate, and finally fructose 1,6-bisphosphate. This 6-carbon fragment is going to be cleaved into 2 3-carbon fragments. Okay. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Right. The enzymes that we're going to encounter are hexokinase, phosphohexosisomerase, phosphofructokinase 1, aldolase, and triose phosphate isomerase. Right. All the molecules are shown here, and we're going to go through each step in greater detail. In stage 2 of glycolysis, we're going to generate ATP. So here we are going to take our 3-carbon fragments and make pyruvate. Okay. So glyceraldehyde 3-phosphate can be used directly. So before we enter the second phase, we're going to talk about how dihydroxyacetone phosphate is converted into G3P. Okay. Then G3P is oxidized and phosphorylated to give us 1,3-bisphosphoglycerate. Okay. This will allow for the formation of ATP, and it's formed through substrate level phosphorylation. Then we make and from that reaction we make 3-phosphoglycerate, which is isomerized to 2-phospho or undergoes an isomerization by a mutase to 2-phosphoglycerate, and then we make some phosphoenolpyruvate, and then we net 2 more ATPs as we make pyruvate. And again, Alright, we're going to go through each of these reactions in greater detail. So remember that the second stage is the payoff phase. This is where we generate ATP and we recuperate the loss from the first stage of glycolysis. Okay, so if we look at the fates of pyruvate from glycolysis, these two molecules of pyruvate, we have three different options for them. We can have them undergo aerobic oxidation, where they enter the tricarboxylic acid cycle and are fully oxidized to carbon dioxide. Okay, this will yield the most ATP. Pyruvate, under appropriate conditions, can also undergo anaerobic oxidation, where we would form two molecules of lactate. This can be seen in contracting muscle. Okay, yeast can ferment pyruvate and make ethanol and carbon dioxide. Okay, again, remember that when pyruvate enters the tricarboxylic acid cycle, that's how we get the most ATP out. In the first phase of glycolysis, we see coupled reactions. Okay? Now this is a theme throughout metabolism where you will couple a reaction to the hydrolysis of ATP to make it favorable. Okay? There are two such reactions in the first phase of glycolysis where they are not energetically favorable, so they're coupled to ATP hydrolysis to drive the glycolytic pathway forward. So when we go through this, you'll see that stage 1 is going to cost 2 ATP molecules. Alright, so the first reaction in glycolysis is the phosphorylation of glucose. And this is accomplished by a hexokinase or a glucokinase. This is a priming reaction. ATP is consumed here. And it is consumed here so we can get more later. Okay? This is going to make the reaction favorable when you hydrolyze ATP. Alright. So you're going to need to be able to interconvert between KEQ and the free energy. And I'll write that equation on the next slide. Okay? Alright, so just to review, the Gibbs free energy under standard conditions is going to equal negative RT ln KEQ. Okay, if we rearrange this, ln of KEQ equals negative delta G over RT, or the equilibrium constant equals E times negative delta G divided by RT. Okay, R equals 8.314 joules per mole per Kelvin. Okay, and T is Kelvin temperature. We looked at this a little bit in Lecture 1, and we will revisit it when we do the homework for this chapter. I will give you this equation and this equation on an exam, and I would also give you these constants. You just need to be able to plug in to the equations. What this table is showing you, it is showing you the reactions and the thermodynamics of glycolysis. Now, as far as we're concerned, glycolysis will end here. Okay. And then if you sum up all of these reactions, you'll get the net reaction that I drew in the beginning, and you would see that delta G is negative. You don't have to memorize this table. I'm just showing it to you so that you can be familiar with these values. Okay, so let's get into the nitty-gritty of each of the steps of glycolysis. So the first step in glycolysis has a large negative delta G. And here hexokinase is going to trap glucose in the cell. And the way it does this is that it's going to phosphorylate the glucose. Okay. Now it says here that I have here that hexokinase is regulated allosterically or by its product, glucose 6-phosphate. Okay. This is not the most important site of regulation of glycolysis. And when we have lecture 3, we'll talk about why, and I'll review allosteric regulation then. Okay, if you can see here, this is the net reaction. We are going to phosphorylate this OH group, and I will show you the mechanism on the next slide. So this is going to be the mechanism of phosphorylation. So here we have our adenosine. This is represented, this would be the adenine and the ribose. Okay. And remember, there are 4 negative charges on ATP. There's a magnesium present with the enzyme that's going to coordinate these 2 oxygens. Okay. Now, the oxygen, I'll draw the glucose. Okay, so the oxygen is going to act as a nucleophile and attack that phosphorus. And then these electrons go out onto that oxygen. Let's draw the magnesium that was coordinated here. Now what happens is we are going to break the oxygen. We're going to have these electrons that are on this oxygen come down, and we are going to break this oxygen phosphorus bond. Now we get a molecule of ADP, because we only have 2 phosphates. Okay, the magnesium is still coordinated here. And now we have, and then we have our phosphorylated glucose. Okay, I did forget to show you up here a base taking off that hydrogen. Okay, so this is our glucose 6-phosphate, and this is the mechanism for the reaction. So this reaction is important for two reasons. Glucose 6-phosphate cannot pass through the membrane because it is not a substrate for glucose transporters. So if you look at this picture here, you can see that when you have glucose in the extracellular fluid, it will pass through the membrane via a glucose transporter. Okay. Once glucose 6-phosphate is formed, it can no longer cross the membrane. So this is going to trap the glucose 6-phosphate in the cell. Second, when we add a phosphate group to glucose, it destabilizes it. This is going to facilitate its further metabolism. Now, the hexokinase that facilitates and catalyzes this phosphorylation is a common theme you'll see in biochemistry, where we have this phosphoryl transfer. Okay? Now, glucose 6-phosphate is common to several metabolic pathways. So once we form the glucose 6-phosphate, it can have several fates. It can go along the glycolytic pathway here. Okay? And make fructose 6-phosphate, and glycolysis will continue. It can be used to make glucose 1-phosphate, which goes on to form glycogen. It can be processed in the pentose phosphate pathway and used to make the synthesis of certain nucleotides. Okay, so I did mention this previously, and we will discuss it in more detail. Hexokinase is regulated, okay, but it is not the committed step. And the reason why it's not the committed step is because glucose 6-phosphate has several fates in metabolism. Okay? So this would be a dot that has multiple branch points. So if you look at X-ray crystallographic studies of the yeast hexokinase, it's been revealed that the binding of glucose induces a large conformational change in the enzyme. Hexokinase consists of two lobes. Okay, you can see them here and here. Now, when glucose is bound, those two lobes are going to move closer to each other, and this cleft closes. So in green here, that's glucose. So you can see the clefts clamping down on glucose. So all of the glucose is encased except carbon 6. And carbon 6, it's the oxygen of carbon 6 that accepts the phosphorylation, or the phosphoryl group from ATP. Here, A is showing you mammalian hexokinase 1. Now, the induced structural changes are significant in two respects. First, when you enclose the glucose, in the enzyme, okay, the environment becomes nonpolar. And so because the environment is nonpolar, this is going to favor reaction between the hydrophilic hydroxyl group of glucose and the terminal phosphoryl group of ATP, because that's sticking out. Okay, remember that C6 is not in the cleft. So that's why this is the site of reaction. Second, the conformational change enables the kinase to discriminate against water as a substrate. The closing of the cleft keeps water molecules away from the active site. If the hexokinase were rigid, a molecule of water occupying the binding site could react with the phosphoryl group of ATP instead of the OH on carbon 6. All of the kinases that take part in glycolysis that we'll encounter, phosphofructokinase, phosphoglycerate kinase, and pyruvate kinase, they all have clefts between the lobes, so that when the substrate is bound, it is able to react with ATP. And this substrate-induced cleft closing is a general feature of kinases. Reaction 2 in glycolysis is the isomerization of glucose 6-phosphate to fructose 6-phosphate, and this is catalyzed by phosphoglucoisomerase. Now, as I said earlier, when we make glucose 6-phosphate, we destabilize it. So the ring is more likely to go into its open chain form. If you look at the open chain form of glucose, there's an aldehyde at carbon 1. This is called an aldose sugar. What we are going to do in this step is we are going to isomerize glucose 6-phosphate to fructose 6-phosphate. Fructose 6-phosphate is a ketose sugar because it has a ketone. The reason why this reaction occurs is because this isomerization is going to activate carbon 3, which I'll circle here in red, for cleavage in an aldolase reaction. In reaction 3 of glycolysis, we're going to have a second phosphorylation reaction. Okay, and this is going to be catalyzed by phosphofructokinase. This is going to be the committed step of glycolysis, where we're going to have fructose 6-phosphate, and we're going to convert it to fructose 1,6-bisphosphate. The prefix bis in bisphosphate means that two separate monophosphoryl groups are present, whereas the prefix di in diphosphate, as in adenosine diphosphate or ADP, means that two phosphoryl groups are present and are connected by an anhydride. You'll see what I mean when we look at the structure. Alright, PFK is highly regulated. We will discuss this in lecture 3 when we look at glycolysis regulation. Okay, so if you look here, we have fructose 6-phosphate. We are going to phosphorylate this hydroxyl group. The mechanism is similar to the hexokinase mechanism. PFK has a similar cleft. Okay, so this is the committed step in glycolysis. You can see there's a large negative delta G. This step is basically irreversible. Okay, this is the second priming reaction. It's going to cost us another molecule of ATP. So if you're keeping track, we are down 2 ATP molecules at this point. Right now we've made fructose 1,6-bisphosphate, which will go along through glycolysis. This does not have another fate. It's targeted for glycolysis and glycolysis only. So this figure is showing you the structure of phosphofructokinase, or PFK. We will see it abbreviated as, and you can see that there are the two clefts here. Okay, in red we have the fructose 6-phosphate, and here in orange, okay, this is the ATP molecule, and that's the gamma phosphate that's going to react. So this reaction occurs in the cleft. Okay, we will talk extensively about the regulation of this enzyme when we look at the regulation of glycolysis. Alright, reaction 4 of glycolysis is where we take our 6-carbon intermediate, fructose 1,6-bisphosphate, and it's going to be cleaved by an aldolase into 2 3-carbon fragments. Those 3-carbon fragments are dihydroxy acetone, or dihydroxy acetone phosphate, can't forget those phosphates, DHAP, and glyceraldehyde. Phosphate, which is G3P. So let's cross this out and write G3P. Here we are going to make a Schiff base, or an imine, in the active site. Okay, this is showing you the overall reaction. This reaction is readily reversible. Okay. And the enzyme derives its name from the nature of the reverse reaction, which would be an aldol condensation. So the reverse reaction is formation of an aldol. Right? And here the forward reaction is cleavage of the aldol. Okay. Even though delta G standard here is positive, the cellular delta G is very close to 0. Here, just to refresh everybody on an aldol. Okay, so an aldol has this generic structure here, where we have a carbonyl, a carbon in between, and then an OH group. And we have reaction between a pi bond and a carbonyl to form the aldol. If you recall, okay, this is an enol, where you have an OH next to a double bond. So if we look at the reaction here and the mechanism of what goes on in the aldolase enzyme pocket, you have a lysine. Okay, that lysine is going to attack the carbonyl. This is why we did the isomerization, okay, of glucose 6-phosphate to fructose 6-phosphate. When this nitrogen attacks the carbonyl, the oxygen can get protonated. Now, we can have another basic amino acid abstract a proton, and we can make an imine, and we will lose water. So here you can see the imine. Right, it's an N double bonded to a carbon. It's also called a Schiff base. Okay, because we're protonated. Now, another basic amino acid can abstract a proton, and we can have movement of the electrons between the carbon and the oxygen, and we can break the carbon-carbon bond. We're cleaving the aldol. Okay? So here you can think about this. Let's look back here. Okay? At the fructose 1,6-bisphosphate. We have an aldol because look, we have a carbonyl, an intervening carbon, and then an OH group. Alright, so we're cleaving the aldol, we're breaking the CC bond. The first product is going to be glyceraldehyde 3-phosphate, which is directly used in glycolysis. Because we now have this lone pair on the nitrogen that's adjacent to the pi bond, what we can do is that the pi bond can abstract a proton, we can have movement of the electrons, and we form the imine again. Okay, or the Schiff base, because it's protonated. Now, the Schiff base can be hydrolyzed, okay, and we release dihydroxyacetone phosphate. Okay, and our enzyme pocket is restored to its starting state. The Schiff base that's formed between the substrate carbonyl and an active site lysine, it acts as an electron sink, and it's going to increase the acidity of that beta hydroxyl group and facilitate, so this is the beta hydroxyl group here. Okay, and it's going to facilitate the cleavage as shown. And so as I said, the glyceraldehyde 3-phosphate, the G3P, can be directly used in glycolysis, whereas the dihydroxyacetone is not. So we need a means to convert the dihydroxyacetone into glyceraldehyde 3-phosphate. So this way we produced 2 3-carbon fragments that we can use to generate ATP. Alright, so in order to convert dihydroxyacetone to glyceraldehyde 3-phosphate, we're going to undergo an isomerase reaction. Okay, and the enzyme that's going to do this is triose phosphate isomerase. So we're going to take a ketone and make an aldehyde. This reaction is readily reversible, and it's driven by subsequent reactions of glycolysis. So as glycolysis continues and the equilibrium shifts, then we can form more G3P from the dihydroxyacetone phosphate. So we're going to look at this mechanism in a little bit more detail. This reaction makes it possible for both products of the aldolase reaction to continue in glycolysis. The reaction involves an enediol mechanism. Okay, we're going to look at a glutamic acid in the active site, and it's so we're going to have a glutamic acid in the active site that's going to act as a general base, and we're going to see that triose phosphate isomerase is a near perfect enzyme, which I'll explain in a little bit. So in the active site of this triose phosphate isomerase, we're going to have a glutamic acid and a histidine. Now the glutamic acid is going to abstract a proton from carbon 1, and then it's going to donate it to carbon 2. Right? However, the carboxylate of the glutamate, or the glutamic acid, is not basic enough to pull a proton away from a carbon. So the histidine is going to assist in the catalysis by donating a proton to stabilize the negative charge that develops at the C2 carbonyl. And I understand I'm just saying these words. You're probably thinking, what is she talking about? We'll see it in a little bit when I show you the mechanism step by step. So let's look at this mechanism in a little bit more detail. Here's your glutamic acid 165 and your histidine 95. Alright, so we're starting out with our ketone, and we're going to make an aldehyde. The first step of the reaction, the carboxylate, is going to abstract a hydrogen. Now, to stabilize the negative charge, we're going to take a hydrogen from the histidine. So now in the enzyme pocket, you can see we have a protonated glutamic acid and a negatively charged histidine. We have an ene-diol intermediate. If you remember from organic chemistry, this is an enol. When you have an OH attached to a double bond. We have two OH groups here, hence the diol. The histidine can now take this hydrogen. As a result of taking this hydrogen, we get a negative charge on the oxygen. The electrons can come down. The pi bond can take the proton. When the pi bond takes the proton now, we have an aldehyde at C1 and an OH at C2. So our isomerization reaction is complete, and the glyceraldehyde 3-phosphate can go on through glycolysis. Now, I said earlier that the triose phosphate isomerase, TPI, is a near perfect enzyme. Okay. If you look at the catalytic power of this enzyme, the enzyme is going to accelerate the isomerization by a factor of 10 times 10 to the 10th. So this is compared to the rate of the uncatalyzed reaction. Now, if you look at the catalyzed reaction, catalyzed reaction over Km, which is the substrate concentration at which a reaction is half Vmax Okay. you'll see that value equals 2 times 10 to the 8th molar per second. Now this is close to the limit of diffusion. What this means in plain English is that catalysis takes place every time that the enzyme and the substrate meet. The diffusion-controlled encounter of substrate and enzyme is thus the rate-limiting step in catalysis. So the rate-limiting step is that substrate coming in contact through diffusion with the enzyme. So therefore, the triose phosphate isomerase is an example of a kinetically perfect enzyme. Alright, so we're now ready to move on to the second phase, or the second stage of glycolysis. I'm just going to recap on the bottom the first stage. Okay, so the first stage, we had glucose, and we net form 2 molecules of glyceraldehyde 3-phosphate, and we lost 2 ATPs. These were consumed. Okay. In the second stage of glycolysis, we're going to harvest some of the energy contained in glyceraldehyde 3-phosphate, and we're going to use that to synthesize a net of 2 ATP molecules. The second stage of glycolysis is going to involve 2 very high energy phosphate intermediates, 1,3-bisphosphoglycerate and phosphoenol pyruvate. They are high energy phosphate intermediates because they can donate a phosphate group so that we can synthesize ATP. So if you remember from lecture 1, these 2 intermediates would be above ATP in the table for phosphoryl group transfer. Alright, so reaction 6 of glycolysis. Okay. We're going to convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, or 1,3-BPG. This reaction is going to be catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Okay? Not only is a product 1,3-bisphosphoglycerate, but we're also going to make NADH. Okay? The mechanism is going to involve covalent catalysis and the nicotinamide coenzyme. So we're going to look at the reaction of NAD+. If you remember, this is one of the electron carriers. Okay, we're going to make a good electron carrier in NADH. Also to note, we all know that arsenic is poisonous. So this enzyme reaction is the site of action of arsenate, which is an anion that's analogous to phosphate. Alright, just to show you the net reaction here, we have our G3P. We have a molecule of NAD+ in the enzyme pocket. We have inorganic phosphate. We are going to make 1,3-bisphosphoglycerate. So what we're going to do is we're going to take this aldehyde, we're going to make a carboxylic acid, and then we're going to phosphorylate that carboxylic acid, and out will come a molecule of NADH. As I mentioned before, this enzyme is where arsenate can interact and cause poisoning. So here, if you look at the structure of arsenate, the arsenic could be replaced with a phosphorus. When we put an inorganic arsenate here, we are going to shut down glycolysis at this point. And so this intermediate can no longer go on to react. That is why arsenic is so poisonous. Alright, so if we were to look at this reaction, we can see it as a sum of two processes, or processes. Okay, first, we're going to have an oxidation reaction where we're going to take this aldehyde and make a carboxylic acid. And the NAD plus facilitates this oxidation. Okay, so the aldehyde, this gets oxidized, and this will get reduced. So when we oxidize an aldehyde, we oxidize it to a carboxylic acid. And we're going to get our NADH. The next reaction when we have the inorganic phosphate is going to be acyl phosphate formation. Okay, we're going to phosphorylate here. The first reaction is thermodynamically quite favorable, and the delta G for the first reaction is approximately negative 50 kilojoules per mole. The second reaction is quite unfavorable, and the standard free energy has the same magnitude but the opposite sign. So if these two reactions simply took place in succession, the second reaction would have a very large activation energy and thus not take place at a biologically significant rate. So we're going to couple these two reactions so that this aldehyde oxidation can be used to drive the formation of the acyl phosphate. The key is an intermediate formed as a result of the aldehyde oxidation that is linked to the enzyme by a thioester bond. And as we go through the mechanism, you'll see this. And as I mentioned when we talked about coenzyme A in Lecture 1, thioesters are high-energy compounds found in many biochemical pathways. So the reaction is going to proceed through a thioester intermediate, which allows the oxidation of glyceraldehyde to be coupled to the phosphorylation of 3-phosphoglycerate. So we're going to have our glyceraldehyde 3-phosphate dehydrogenase enzyme. Okay. The G3P is going to bind. A cysteine is in the enzyme pocket that has a thiol, and here is our NADH, our NAD plus in the enzyme pocket as well. The cysteine will attack the carbonyl. The electrons then go out onto the oxygen. As a result, the NAD plus can abstract a proton and we can reform the carbonyl. But now, instead of an aldehyde, we have a thioester. Okay. What happens is the NADH will leave the enzyme pocket. It is then replaced by a new molecule of NAD+, because NAD+ is what remains bound to this dehydrogenase enzyme. Inorganic phosphate comes in. It will react with the thioester. Okay, so instead of reacting with the carboxylic acid, as I showed you on the previous slide, it's reacting with the thioester, which makes this reaction favorable. And now we have 1,3 bisphosphoglycerate. The replacement of the NAD plus is very important. When the NAD plus comes, a new molecule of NAD plus comes into the enzyme pocket, the positive charge is going to polarize the thioester intermediate, and this will facilitate the attack by the orthophosphate. Alright, this example illustrates the essence of energy transformations and of metabolism itself. We have energy released by carbon oxidation, and it's converted into a high phosphoryl transfer potential molecule, which is 1,3-bisphosphoglycerate. So this slide is showing you the favorable oxidation and the unfavorable phosphorylation reactions that are coupled by the thioester intermediate. So if you look at A here on the left, so A is a hypothetical case where there's no coupling between the two processes. So you can see that there's a very large activation energy to overcome in the acyl phosphate formation. Okay. If you look at B, which is what actually happens biochemically, okay, the two reactions are coupled through the thioester intermediate. So this greatly is going to decrease the activation energy for the reaction and allow it to proceed. Alright, now we come to reaction 7 of glycolysis. Here we're going to synthesize ATP from 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is an energy-rich molecule with a greater phosphoryl transfer potential than ATP. So thus this molecule can be used to power the synthesis of ATP from ADP. We're going to see this is a substrate level phosphorylation. Here we're going to pay off the ATP debt that we created in the first stage of glycolysis. If you remember, in the first stage of glycolysis, we cost ourselves 2 ATPs. Here, because we have 2 1,3-bisphosphoglycerates, we're going to get 2 ATPs out of this reaction, 1 per bisphosphoglycerate. And as a result, we're going to settle the score and be at a balance of 0 ATPs. And we will see that 2,3-bisphosphoglycerate is... We're actually going to ignore the 2-bisphosphoglycerate for now and come back to it a little bit later. So this slide is just showing you an overview of the reaction. What we're going to do is you're going to take a phosphate group from the bisphosphoglycerate and we're going to phosphorylate the ADP. As I said before, the kinases typically have the 2 clefts, and you can see that here when we look at the phosphoglycerate kinase, it's showing you the intermediates coming into the cleft and then the cleft closing. Alright, just to show you what this reaction looks like again. This is the phosphate group that is transferred. All right. We have our ADP here. Okay. in the presence of the kinase. Okay, the reaction is reversible too. We also need magnesium. So this is going to be similar to the other phosphoryl transfers that we've seen. We get our new molecule of ATP. In addition to the ATP, we have now formed this molecule here, which is called 3-phosphoglycerate. Alright, let's recap where we are with stage 2 of glycolysis. Okay, so we started out with glyceraldehyde 3-phosphate. We had a dehydrogenase reaction, followed by phosphoglycerate kinase. Now, the net result of this is that the glyceraldehyde 3-phosphate, which was an aldehyde, after these two reactions has been oxidized to 3-phosphoglycerate. Okay. We had an NAD plus and we converted it. It got reduced to NADH. This is going to be used to carry electrons for us to oxygen. And we formed ATP, okay, through ADP and PI. And this was at the expense of carbon oxidation. So I should have stated this in the beginning, but glycolysis is a catabolic pathway, and we're oxidizing carbon. We're oxidizing carbon to produce ATP. And also, keep in mind that we made 2 molecules of ATP because we have 2 molecules of glyceraldehyde 3-phosphate in the second stage of glycolysis. Now we come to reaction 8 of glycolysis. And here what we're going to do is we're going to have another type of isomerization, but this isomerization is going to be done by a mutase instead of an isomerase. So we're going to have a rearrangement. So the position of the phosphoryl group is going to shift, where we convert 3-phosphoglycerate into 2-phosphoglycerate. Now, the reason why we have a mutase is because it's migration of a functional group within a substrate. The reason why this reaction occurs in glycolysis is because it repositions the phosphate so we can make phosphoenol pyruvate, also known as PEP. So this slide is just showing you the net reaction here, where we're going to take the phosphate group that's on carbon 3 and move it to carbon 2. Here we're going to look at the mechanism of this reaction. And it's an interesting mechanism because the phosphoryl group is not simply moved from one carbon to another. In the enzyme pocket, 2,3-bisphosphoglycerate is required. The reason why a small amount is required is because it maintains the phosphorylation of this histidine. We have a phosphohistidine in the active site. So what happens is that this phosphate gets transferred to the 3-phosphoglycerate, and we actually make 2,3-bisphosphoglycerate. Then the histidine will remove the phosphate from carbon 3. So now we have regenerated our phosphohistidine, and now have a histidine on a phosphate, on the second carbon here. Let me just recap this again. We have a phosphohistidine. We are going to have attack on the phosphorus by the oxygen on carbon 2. We then make 2,3-bisphosphoglycerate. Forgot the B. Then the histidine will attack the phosphorus. We regenerate our phosphohistidine in the enzyme pocket, and now we have our 2-phosphoglycerate. Alright, and the next reaction, reaction 9, is going to be catalyzed by an enolase. We're going to take this 2-phosphoglycerate and make phosphoenol pyruvate. The reason why we're going to make PEP is because it has a higher phosphoryl group transfer potential, and this is going to prepare us for the synthesis of ATP in step 10 of glycolysis. The enolase catalyzes this formation of the phosphoenol pyruvate because, as I said, this molecule here has a higher potential for transferring this phosphate group. The reason is that the enol is less stable than the 2-phosphoglycerate. So here you just have a phosphate ester of an ordinary alcohol. If you remember, there was just an OH here. Now we have a phosphate ester of an enol, and this is much less stable, so therefore it will transfer this phosphate group. Alright, so now finally we're coming to reaction 10 of glycolysis, and this is catalyzed by pyruvate kinase. What happens is that the PEP is going to transfer its phosphate group to ADP. We will have an enol that will then tautomerize to form the ketone pyruvate. This tautomerization from the enol to the ketone is what drives these last two steps of glycolysis, reaction 9 and reaction 10. Just to show you the tautomerization when we lose the phosphate group, okay? So when we lose the phosphate group, we will form pyruvate This is the enol form of pyruvate. It will tautomerize to the keto form. And if you remember from the beginning of the lecture, pyruvate can then go on to the tricarboxylic acid cycle to generate even more ATP. So we generate an ATP when we lose the phosphate group here and make pyruvate. Okay, so we've completed glycolysis. Now, if we, again, I'm just going to write down the net reaction we had glucose, we had 2 NAD pluses, we had 2 ADPs, and 2 inorganic phosphates. We made 2 molecules of pyruvate. We made 2 NADH molecules, 2 protons, two ATPs, and 2 waters. The reason we get 2 out is because in the second stage we have 2 molecules of the glyceraldehyde 3-phosphate, that G3P. Now, there were 3 chemical transformations that occurred. I know it seems like there were more. Okay, we had the degradation of the carbon skeleton, where we went from a 6-carbon glucose to a 3-carbon pyruvate. Okay. We had phosphorylation of ADP to make ATP, and this was done by compounds with high phosphoryl group transfer potential and 3, we had transfer of hydride to NAD plus to make our carrier NADH, which is going to carry the electrons to oxygen. The overall delta G for glycolysis is negative 85 kilojoules per mole. This is going to conclude our discussion of the glycolytic pathway in the forward direction. We will talk about regulation and its role in cancer in Lecture 3. Thank you for sticking with me.